okay? So it's... We're still on time. Shocking. You know, I planned everything perfectly that the Cures Act was going to pass the Senate today, and carefully collaborated with everyone in Washington just because I knew Dr. Whitten was going to be here today. And I'm just making a little joke, okay? It is really, we're really honored to have Celia Whitten, who's dedicated so long and made so many efforts on behalf of making sure that cell therapies are safe and moving forward. This is a very special session, a very timely session to learn more about the FDA and what it does in our field, and also hear from an attorney and a representative of industry to get a full perspective of where our field is going. So I'm going to call up to introduce her panel, our friend Dr. Julie Alexson of the Wake Forest Institute for Regenerative Medicine.
Thank you, Bernie. Good morning to everybody. We're excited to be here today to talk about the FDA oversight of regenerative medicine and how can we accelerate to the clinic, really critical. As Bernie said, we have three very distinguished speakers this morning that are with us, and we're honored to have Dr. Whitten from FDA, Mr. Shineson as a legal perspective, and Mr. Martin McGlynn as the industry perspective, and I will introduce them in just a bit. But just want to say that there's been quantum leaps in regenerative medicine, and I am really excited to see the movement forward, although it's definitely a little slower than we'd all like to see. But a lot has been happening, especially in immunotherapy, leaps and bounds, gene therapy, CRISPR. A lot of things are coming in that we're looking at being able to move into the clinic, but I think the immunotherapy definitely is working on a fast pace. So that's some of the positives. As we look at tissue engineering, we have some huge national efforts that are moving forward, such as AFIRM-2, looking at moving forward with tissue engineering, many different areas. It's a $75 million effort over 5 years with 30 organizations and 60 projects, so that's huge. We are also involved, Wake Forest, in Body on a Chip. We're hoping that that's helping in the field of diagnosis, as well as maybe looking at scaling up for organs and tissues. So these are two large efforts involved in advanced manufacturing. The White House wants to see that go forward. That would be huge in the area of the health economy. We really need to bring the manufacturing here back to the US, which would be good for us. And as we look at the cost, there's been several studies done by Tufts University in 2014 and then recently in 2016. And as we look at a comparative analysis for drug development, they're looking at about $2.6 billion costing to be able to move the product to the clinic and market. And that's pretty steep, with about $1.4 billion out-of-pocket costs, and then another $300 million post-market. So I really think, as I said this morning, there's got to be a way that we all work together to be able to lower those costs. We have a translational team at Wake Forest Institute where we try to accelerate as much as possible, work closely with the faculty and staff. We have over 450 faculty and staff, and kind of a unique way to be able to accelerate, but everything can be done under one roof to move it forward. So I think that these are some of the highlights in regards to moving it forward. This just shows you how long does it take. Everybody asks me that. How long will it take to get something to clinic? Anywhere from 7 to 15 years. And we really want to be able to scale that back if possible. So I like to show this example of diabetes, and that we're actually spending about $300 billion annually, which is huge. And if we could find that cure, this would look very different. So healthcare costs decrease, and then the quality of life. So there's been a lot of efforts. The Diabetes Research Institute's making extreme headway in this field, so we're really excited to see things move forward. Just wanted to show you, this is exciting because we see over 700 companies in the regenerative medicine space. This was put out by ARM, Alliance for Regenerative Medicine, their Q3 data report, and then we're seeing 800 clinical trials. So this is mainly in the cell therapy is what they were looking at, but what's exciting is that we're seeing the large amount in phase 2. So a couple years ago, you'd have seen a higher number in phase 1, so this is really exciting for the field. So it is moving forward, but definitely not as fast as we'd like to see. So with that, I'd like to introduce our speakers this morning. We have Dr. Celia Witten. She is currently the Deputy Director of CBER, Center for Biologics Evaluation and Research, which is a newly appointed position in 2016. She was Director of the Office of Cell Therapy, Tissue, and Gene Therapy since 2005. Between 1996 and 2005, she served as Director of the Division of General Restorative Neurological Devices in the Office of Device Evaluation in the Center of Devices and Radiological Health. She's a board certified in physical medicine, rehabilitation. Her BA degree was earned at Princeton University, and prior to attending medical school, she was at the University of Miami, and she received her PhD from Stanford University. So I'd like to welcome Dr. Witten to the stage. Thank you.
Okay, thank you. Can you pull my slides up? Oh, good. Okay, first I'd like to thank the organizers for inviting me to speak at this meeting. I'm going to speak about the Center from the perspective of the Center for Biologics. As Julie mentioned, I'm the Deputy Center Director, but until recently it was Office Director of the Office of Cell Tissue and Gene Therapy. I'm going to talk somewhat about the office itself, the center of the office, some of the things that are happening in the center and in the office to address new regulatory needs. Is there any way to turn that echo to stop echoing? Maybe not. Okay, so as Julie mentioned, just one basic fact, which is the Center for Biologics is one of 3 centers that are responsible for medical products at FDA. And one of them is the Center for Drugs, and another is the Center for Devices, and then there's the Center for Biologics. And as Julie mentioned, prior to my 10 years at CBER, I spent 10 years in the Center for Devices. I'm not going to read this, but these are the kind of products that are evaluated at the Center for Biologics. And you can see they span a range, but they include, in addition to the products in cell tissue and gene therapy, blood products, allergenics, and vaccines. So one recent change that I'm not sure if people are aware of is that there was a reorganization at the Center for Biologics recently that affected the Office of Cell Tissue and Gene Therapy. And what happened is that certain review functions were consolidated into one, and that consolidation consisted of all of what people know of as OCTGT and part of what was formerly the Office of Blood Regulation and Research. So I have indicated in this slide what part of OBR are transferred into this new office. And as I mentioned, OCTGT is in toto as part of OTAT. A few other changes that I don't have on this slide in the Office of Tissue and Advanced Therapies. So the current newly appointed office director is Dr. Wilson Bryan, whom many of you may know from his role as the division director for the Division of Clinical Evaluation and Pharmacology Toxicology in OCTGT. And also there's some new division management. The Division of Human Tissues Division Director, Scott Brubaker, who joined us recently from AATB. So I'm going to move on and talk about breakthrough therapy, which is a program that's a recent program, and it's gotten a lot of interest, and it's a pretty exciting program. This program was signed into law a couple years ago as part of FDASIA, And the intent is to expedite the development and review of a drug intended alone or in combination with one or more other drugs to treat a serious or life-threatening disease or condition, and where there's preliminary clinical evidence that indicates that it may demonstrate substantial improvement over existing therapies on one or more clinically significant endpoints. So this program applies to the products in CEDAR and also CBER. There's a guidance that describes the criteria for this. People get confused, including people at FDA, I would say, because they're initially, when it was enacted, because there are so many different programs for expedited or accelerated development. So there's a guidance that consolidates the information about all of these programs, which includes fast track, breakthrough, accelerated approval, and priority review. And there's a nice table which describes the features of each of these and how they're, and it allows you to compare all at once. But breakthrough therapy is what I'm going to focus on, and the breakthrough therapy gives the sponsor intensive guidance on development as early as phase 1, with an organizational commitment involving senior management. There's been a number of these breakthrough therapies granted in what was the Office of Cell, Tissue, and Gene Therapy, and I think the intense development help that sponsors receive early on in development, which is how it has worked out in former OCTGT, has really benefited the sponsors and also FDA because it allows us to do planning in advance. So what happens with breakthrough is that we ask the sponsor to develop a communication plan that lists what they consider to be all their milestones, and we arrange for meetings and discussions of what those milestones are, how to achieve them, and what the goals are. So there's more frequent and regular meetings than the other programs allow for. So this is just a history of how many breakthrough therapy designations. This is CBER in the whole. The ones in the dark blue and the purple are the ones from what was OCTGT and is now part of OTAT. So it gives you an idea of how many have been approved. So it's a fair number, and like I say, it's an exciting program, and I think that we've received enthusiastic feedback from the sponsors, and certainly as office director, I received enthusiastic feedback from the staff as well. So people are always asking me why anything gets turned down for breakthrough. So these are some of the reasons from OTAT, which is evidence is too preliminary, not really interpretable even to figure out what a signal would be, post hoc analyses of failed studies and identifying a subset. Some of them don't seem to show improvement over available therapy, or it's really a completely different product or indication from what the sponsor has evidence on, something completely different from what their application addresses. So next I'd like to move on, and as I mentioned at the beginning, I want to talk about what's our approach to preparing ourselves or addressing problems in the future. Certainly addressing emerging infectious diseases is a challenge for the center as a whole. When there's a new emerging infectious disease, when I talk about that, I'm talking about Zika, Ebola, that kind of thing. The center as a whole, because of the types of products that it reviews, has a center-wide response of dialogue and communication and trying to assess what the issue is and how it affects the operations of the center. And here would be 3 different product office-specific responses. So obviously, vaccines, preventive vaccines, safety of the blood supply, safety of human tissues, those are all important. So here are some specific examples of the response, and this is from old OCT, I call it OCTGT, now part of OTAT. So West Nile virus, recent guidance, and this guidance is different from previous guidances in that it delineated between living and non-living donors, and also between seasonal, and also allowed for seasonal testing. And that was, I think, in recognition of the fact that this was the most practical and adequate to reduce the transmission by HCTPs. but we'll continue to follow what current studies there are so that we'll have a better understanding of transmission. Zika virus is another recent example where the guidance document for tissue transplantation made a distinction, had differential screening recommendations for living donors, gestational tissue donors, and cadaveric donors. And that's because on information at the time, it appeared to be that the types of cells and tissues with highest potential for transmission appear to be recovered from living donor. There's less evidence for the types of tissues recovered from non-living donor and their potential for transmission. But again, there's a recognition that the risks may evolve as more information is available. But what I want to mention in specific, in light of the consideration I mentioned about being prepared for future needs and future considerations, is that the center also recognized the need for additional scientific information to understand the risk of transmission of Zika virus by human cells and tissues. And we were fortunate to be able to initiate 2 studies to evaluate Zika virus in non-human primates. One is at UC Davis to look at samples during initial evaluation of non-human primate model of Zika disease, and the other is to look at tissues that support replication of Zika virus and duration of Zika viral RNA in body tissues during pregnancy in a non-human primate. I mean, these kinds of studies are important because what is not known really is, so we're asked, well, why can't you use blood testing to look at safety? And the fact is, I think we need an understanding both about the blood tests and what they would or wouldn't tell us about tissue safety for various tissues. So hopefully these studies will elucidate some of that. Now I'm going to move on to some specific topics in OTAT. One is gene editing, and this isn't going to tell anybody what to do with their gene editing technology. It's just to let you know what we are doing to, prepare for oversight of this area. So what is gene editing? Probably most of you know. It's DNA, and otherwise I think there are some longer sessions in this meeting. DNA inserted, deleted, or replaced in the genome of an organism with engineered nucleases. This field has a lot of potential to modify somatic cell or germline genomes through relatively efficient targeted genetic modification. And we regulate currently somatic and germline gene modifications used as therapeutics in humans. This includes modifications of cells prior to administration and gene therapy vectors. And at the moment, I think the somatic cell therapy is what people are mostly talking about in humans. So what are some of the scientific considerations? Well, they're very similar to what we would consider scientific considerations for gene therapy as a whole, although there's, additional issues regarding how you would evaluate it for gene editing versus other kinds of gene therapy. You want a mechanism that has efficient delivery on target modification, targeted expression, and you may want it to be long-lasting depending on the indication, and you want to avoid off-target editing, off-target expression, and insertional mutagenesis. And of course, there's always the consideration for potential germline transmission. So we look at this like we look at other gene therapies in terms of the nature of the editing, whether it's inactivation, insertion, or modification. Safety considerations would include percentage of cleavage at on and off target sites, and we'd evaluate the profile of the insertions and deletions and mutations generated. And we look at this in a science-based approach and look at benefit-risk analysis. So how do we build capacity at CBER for review of these kinds of new technologies? Well, we have two new principal investigators who were recruited, so they work in the lab and in the review organization to address emergent scientific needs in this area. We participate in public meetings and workshops on gene editing, participate in the recombinant DNA advisory committee process with NIH, where some of these can be presented publicly. We participate as a non-voting member. We have continuing workforce education, and this was sponsored by the Health Research Alliance. We had a recent workshop with some really, truly excellent speakers earlier this year in November. We've co-sponsored a study with the National Academy of Sciences to study gene editing technologies to provide a framework on principles that can be adopted and adapted by any nation considering the development of guidelines. So those are some of our activities in that area so that will be prepared. We have some other specific activities. I want to highlight one of them because I think it is quite exciting. The Kidney Health Initiative was a public-private partnership founded in 2012 by the American Society of Nephrology and the FDA. And the leadership for this, I will say, is in the Center for Drugs. So they really get a lot of credit for stepping forward to put this initiative together with ASN. But we're fortunate to be able to participate in it, both in terms of their activities and also I'm privileged to serve on the board. So who are the members of this group? Patient and health professional organizations, commercial entities, research institutions, and federal agencies. And the types of projects are collaborative and member-driven. I'm going to talk about one specific project that was driven by the Center for Biologics and the Center for Devices. And this is called, the formal name of it is Development of a Roadmap for Innovations in Renal Replacement Therapy. It's a joint project by our center and CDRH. The understanding is that innovative technologies could potentially increase renal replacement therapy options for patients. And the goal is publication of a roadmap to allow innovators to understand challenges and milestones along the path to successful development. So the status of this project is there's a project plan with the steering committee and plans for a public workshop, work teams, and a time frame for deliverables. If you want more information on this, you can look on the website for the organization. I'm going to just talk a little bit about bioprinting, and again, just to answer the question, what do we do to prepare for this? So just to put everybody on the same page, there's subtractive manufacturing and additive or 3D printing, of which 3D printing is a type. And I think, people probably know what this is. There's an FDA additive manufacturing working group, which is led in CDRH, but we CBER participate in it. It's had a number of activities. The first is this public workshop. The second one is actually a CBER workshop for bioprinting. There's a joint draft guidance in 2016, but I want to mention that in terms of preparing for the future, we have commissioners' fellowships in regenerative medicine. So one of the commissioners' fellowship topics, or that we recruited for, was for someone with expertise in this area, and her project was to analyze trends and regulatory concerns for these devices and biologics, which she's in the process of preparing as a paper. And we were fortunate to be able to hire her as a full-time reviewer. One other thing I'll mention is the National Academy of Medicine Forum on Regenerative Medicine, which we co-sponsor with a number of other organizations. It serves as a neutral convening mechanism for interested parties, and it allows an opportunity for dialogue around opportunities and challenges for translation of regenerative medicine to marketed products. The kickoff meeting was last June, and there was a workshop in the fall, and based on what we learned at the workshop, there's a number of working groups that are now formed and starting their work. I'm not going to spend too much time on these tissue guidances. I'm aware that the next speaker or one of the following speakers is going to talk about them and his views in more detail. But I will say that we published 4 guidance documents in response to the need that we saw for further clarification regarding the tissue rules. These were published, and the titles are here on the slide. I won't read them, but we reopened the comment period since they were published at different times in draft. We reopened the comment period and had a public Part 15 hearing in the fall, and then subsequently closed the comment period, and are in the process of reviewing the comments with an aim to finalizing the guidance. And this is oh my gosh, sorry. Did not realize this wasn't advancing. There are the guidance documents and the Part 15 hearing. There were about 95 speakers at this Part 15 hearing. I want to talk about some ongoing priorities that we have. The first one is standards activities in regenerative medicine. This isn't new. We've had liaisons to various standards development organizational technical committees for some time. There's a standards working group internally where we review our participation and decide what are our priorities. We have sponsored and participated in workshops. Our lab has a number of activities in the standards area, including round-robin testing, looking at test methods, and we have other collaborations with NIST also. And we're certainly interested to see the progress of the standards coordinating body that was stood up by the Alliance for Regenerative Medicine, and hoping to have active participation in that. Here are some of the committees that we are active in. Some of you may be familiar with these and may be participants. We're also often asked what we do internationally. We have a number of activities that support international regulatory convergence. There's a group called the International Pharmaceutical Regulators Forum, which is something that happens around the margins of ICH. It's not as formal as ICH, and it allows more flexible participation and discussion. We have an agreement with Health Canada EMA in Japan, and we have a cluster meeting on advanced therapy medicinal products that takes place. And there's some other cluster meetings that also touch on regenerative medicine products like pediatric cluster, oncology cluster, that we also participate in. We offer sponsors the opportunity for parallel scientific advice. Some have taken advantage of this opportunity, and when they have, I think it's been extremely productive. We participate in the APEC Regulatory Harmonization Steering Committee. We've had personnel exchanges between regulatory agencies, and we also have a confidentiality agreement with HFVA, which was primarily around the topic of the mitochondrial replacement type of technologies that have been developed. There are other activities. I don't have time for any detailed slides on these, but I just will say there's a number of guidance documents from OTAT, aside from the ones related to tissue. There's also a website called OCTGT Learn, where sponsors or others who are interested can access a wide variety of talks around regulatory topics that affect OTAT. The science of patient engagement. Patient engagement has been a big topic, and I don't have a number of slides on this, but I just will mention that the patient input meetings that have been had actually are co-sponsored by CDER and CBER. CDER's the lead on most of them, but CBER has led a couple and will be leading one in 2017. But we have other activities related to patient engagement as well. And some patient groups have asked to meet with us. Some have asked for input on their patient preference studies. We certainly invite patient reps to a number of our advisory committees. So we have some activities in patient engagement. I'd be happy to answer questions specifically on the patient preference studies, which I didn't include in this talk, if there are any, during the question and answer session. We have research activities, and I've just highlighted two. One is on quality of life measures for Wiskott-Aldrich syndrome by one of our researchers, Rob Sekulik, and the other is the laboratory research in the area of gene therapy. And these are just examples. There are certainly others. So I mentioned that we have a number of guidances in the cell and gene therapy area. I have this list here at World Stem Cell posts the slides. You can access this list from the slides. These are recent tissue donor eligibility guidances, which include the ones that I mentioned in my talk, plus a couple others. So what challenges do we see coming up? Well, there's obviously implementation of user fees is always an ongoing activity, and there may be other new legislation. Obviously, I can't comment on legislation that's under development or under consideration, but we are certainly prepared to work on any implementation measures that we need to for anything that gets passed. So I just want to say in closing that we in CBER are extremely excited about stem cell therapies in general. I think they hold a lot of promise. We are excited about working with sponsors. We think that there may be some, really great advances in the next couple of years. There's considerable flexibility in our regulations as it stands in interpretation to be able to work with sponsors to help them develop their treatments. And just to say that we certainly want to make sure that these are safe and also that they deliver value and that they're effective for patients, which I know is the goal of everybody at this meeting. Thank you.
Thank you, Dr. Witten. We're hoping to have a few minutes at the end to be able to allow for questions. So our next speaker is Mark Scheinison. He's the partner in a Washington, D.C. office, Elston & Bird. He heads the firm's food and drug law practice. Mark previously served as the Associate Commissioner for Legislative Affairs at FDA. He came to D.C. to work as a legislative assistant and counsel to Representative Bill Gradson, the ranking member of the Health Subcommittee of the House of Ways and Means Committee. In addition, he worked as a regulatory FDA lawyer for another national law firm, senior vice president, Ketchum Communications, and principal in government relations. He received his BA and his JD from the University of Cincinnati and his college, the College of Law, and his LLM from Georgetown University Law Center. So I'd like to welcome Mark Scheinison.
Thank you, Julie. Good morning. It's an honor to present to this group of industry pioneers. I'm going to start with the 12 words you might fear most, which is, I'm a lawyer from Washington, D.C., and I'm here to help you. So these aren't my slides. What do we do next? Oh, here we go.
Yes. Can you change the slides, please?
Slide should be from Alston and Bird. My worst fear with technology. As these are being changed, I will report that.
I'm sorry, is there a problem with the slides? Okay, you know what? Do you mind if I switch while we get your slides up? I'm sorry.
No problem.
I think it's better because, or would you rather just?
Howard, do they have my slides?
They do. They have them. I checked. Okay, so we're going to just switch up while we get Mark's slides. And so I'm going to introduce Martin McGlynn. He's a stem cell therapy pioneer, former president and CEO of Stem Cells, Inc., and now a board member of the Regenerative Medicine Foundation. Mr. McGlynn served as the CEO, president, and director of Stem Cells, Inc., as the company developed applications for cell therapy platform technologies in multiple therapeutic areas and brought neural stem cell therapy in cervical spinal cord injury into a phase 2 clinical trial. He's held multiple positions in leadership, nonprofit advocacy groups in the area of regenerative medicine. His global experience includes roles in Becton Dickinson, Abbott, Anaquest, and Pharmagen. He has served as the member of the board of directors of the Confederation of the Irish Industries, as well as board of the Pharmaceutical Manufacturers Association of Canada. Mr. McGlynn is a graduate of the University of College of Dublin and pursued advanced studies at the Irish Institute of Industrial Engineering and the University of Birmingham, England. I'd like to welcome Martin McGlynn.
I thought I was going to get away easy, hoping Mark would give my talk. Good morning. I'm here in two capacities, as Julie has indicated. I recently had a great privilege and honor to be invited to join the board of the Regenerative Medicine Foundation. I like to call it Bernie's board. I recently retired as Chief Executive Officer of publicly traded biotech company Stem Cells, Inc., whose scientists isolated and discovered a very rare population of expandable human neural stem cells and published in PNAS in the year 2000. I joined in January of 2001 to help lead the effort to translate this fantastic discovery into cures and treatments for degenerative diseases and serious conditions of the central nervous system. I think, from my perspective, I absolutely concur with the notion that we need to do great science. And we need to design and conduct excellent clinical trials to tease out the benefits as well as the risk profile for cell therapy. But we also have to do something else, and that something else is we have to come up with a sustainable business model to help make this happen. Typically, phase 1-2 studies are used to tease out the pivotal studies and the approval studies, tease out the kinks, figure out what works and what doesn't work. In the ecosystem of sponsors in the corporate world, we live or die by early studies, typically open label studies. And this is what is known in the industry as the valley of death. I can attest to the fact over 15 years, Stem Cells Inc. went through many near-death experiences, and in the end, one proved fatal. Many sponsors do not make it beyond open label studies, so the existence of other enabling mechanisms at the later stage of the development runway are moot in most cases. And the paradox we have is that potential strategic investors in the pharmaceutical industry typically will not engage without compelling phase 2 controlled data. And that is the crux of the matter for the translator sector in regenerative medicine. For the purposes of this talk, I went back and I took a look at a representative sample of companies that I would describe are pioneering cell therapy companies. This is not meant to be an exhaustive list, but a true representation. And I apologize to anybody whose name or whose company is not on the list, but it's meant to be illustrative. There's about a dozen companies on this list, and I think when we go through this, you'll agree that the track record has not been all of that fantastic. You have to go back to 1988 to the first effort to industrialize cell therapy with the hematopoietic stem cell discovered by Irv Weizmann at his lab in Stanford that led to the creation of Systemics in 1988, which in 91 was acquired by Sandoz, became Novartis. And that was closed in 1999 when the owner of the technology decided that a patient-specific business model just did not fit with their role as product developers in the pharmaceutical industry. In 1989, Astrum was established to develop autologous cell therapy. They ran into a number of serious challenges. They're continuing to work with autologous therapy, but they decided strategically to refocus their efforts in 2014, and they acquired a product business from Sanofi, which had acquired Genzyme. And Sanofi decided that this particular business was not of interest to it, so Astrum picked up those products. And then you move to 1990s when the pace accelerated, and you had Geron, which was created in 1990. Furthest they got was to open label studies in spinal cord injury, and they ended up selling their assets to Biotime in 2013. Osiris was established in 1992 after some tremendous work done by Dr. Arne Kaplan. The furthest they got was they got approvals for these products for graft versus host disease, I believe, in Canada and New Zealand. And they ended up selling their assets to Mesoblast in 2013, an Australian company. In 1992, Dendrian was established, and I think most people know that they managed to get Provenge approved in April of 2010. This is using patient-derived dendritic cells in cancer. They ended up in bankruptcy in 2014, and their assets were acquired by a famous company called Valiant. In 1994, ACT, which changed its name along the way to Alcata, were developing RP cells derived from an embryonic platform. They were established in 94. Furthest they got was open label studies before they sold the company to Astellas, a Japanese company. And then Neuralstem was established in 96 to develop human neural stem cells. They managed to get to open label studies in ALS, and they exited the activity in 2016. Athersys was established in 96 to develop multipotent adult progenitor cells. This was work developed, created by Catherine Verfeil. And Athersys has hit a number of speed bumps along the way, but they are fighting the good fight, and they continue to do clinical trials. Their leading product is in stroke. They are in phase 2. The company that I led for 15 years, Stem Cells Inc., established in 97, discovered human neural stem cells and published that information and data in 2000. The furthest we got was with open label studies, 2 in the brain, 1 in spinal cord, and 1 in the eye, and early stage phase 2 data when, for various reasons, the company decided to wind down activities and sold the neural stem cell technology to a Chinese company called Bright Oceans Corporation. Reneuron was established in 97. Notwithstanding that, they were also developing c-MYC immortalized stem cells, neural stem cells. They're still in the game, but they're in very early stage clinical trials, and they continue to work that technology. Cytori, which was established in 2002 for adipose, patient-specific adipose cells derived from adipose, I should say. And they're still in the game in phase 1-2. And last but no least, Mesoblast, which is an Australian company. It's probably the largest and most well-established and funded cell therapy company that we know of today, established in 2004. They are leading the field in phase 2, phase 3 with mesenchymal stem cells, and they've managed to get a BLA approved in Japan for graft versus host disease with their Japanese partner. I think in some respects this is a depressing picture in many ways. However, I do think that, like everything else, the pioneers are not only those who are hanging over the barbed wire fences with the arrows in their backs, but they are truly the necessary element that's a prerequisite ultimately to breakthrough therapies. Take-home message. There's a definitive time limit to the market's patience waiting for compelling phase 2 data in this space. Translation efforts are taking way too long. 15 to 20 years is not unusual. Autologous cell therapy and big pharma, at least in the N=2 analysis that I've shown you today, are not good fits. Patient-specific product approval are not a guarantee of a profitable business. Exhibit 1 would be Dendrion. Non-US entities, however, recognize value in this space and are continuing the efforts. ACT or Okada acquired by Astellas, a Japanese company. CDI was acquired by Fujifilm. This was a company established by Jamie Thompson. The technology from Stem Cells Inc. was acquired by Boco in China. Seska, another California company, acquired by BioLife in China. Osiris' expanded cell culture mesenchymal cells were sold to Mesoblast. And Helios, another Japanese company, has partnered with Athesis and Universal Cells. So the battle continues. One of the biggest challenges I think we're going to face, and my co-panelist, Dr. Celia Whitten, biology, proteomics, genomics, epigenomics, immunology, and bioinformatics, just to name a few, will present challenges of a kind that have not been seen to date in the relatively simple world of unmodified cell therapy. The 21st Century Cures Act, which I believe is poised to come into law, is a good start. It brings regenerative medicine in out of the cold, and it formally acknowledges that it needs our full attention and support. And it gives the FDA additional tools that I hope they will put to good use. Thank you.
Sorry about the switch around. So I'd like to ask Mark Scheinison to come to the stage. Thank you so much. Thanks, Mark.
So these are really great slides, and this is just a setup that I'm selling them in the back of the room if you'd like them after the presentation. So while we're waiting, the 21st Century Cures, as you know, has a title on regenerative medicine. I will detail some of what it contains, and happy to answer questions during the course of the conference. I'll be here for the next couple of days. That legislation, as you know, passed the House last week by a vote of 85 to 13. I'm sorry, by a vote of 392 to 26, and the test vote in the Senate yesterday to invoke cloture over the objections of Senator Elizabeth Warren passed 85 to 13. So sometime in the course of the day, this is going to be passed as one of the few last contributions of the Obama administration, I guess, and the Congress agreeing to a bipartisan compromise. It's necessary because, as many of you know, the Food, Drug, and Cosmetic Act, which was, thank you, which was enacted in 1938 originally, believe it or not, never heard of cell therapy. And that legislation is directed to drugs, biologics, and devices, but there's no statutory basis for the regulation of cells or cellular tissue. A case, Regeneration Technologies, that you may be familiar with, reinforced FDA's jurisdiction to regulate cells as drugs. And the drug paradigm just hasn't worked. It's created sort of a Grand Canyon where you have regulations under 1271. Okay, good. Let's see. Okay, so as many of you know, human cell tissue products were created by regulation, not statute, and there are completely separate regulatory regimes, thank you, Julie, for organs and cord blood and secreted or extracted human products and manipulated bones and animal cells and in vitro diagnostics. It's a very complicated regulatory paradigm that this statutory change is intended to address. So some therapeutic it's kind of like the three bears when they got to the furniture. Some regulation is too soft and some is too hard, and we're just trying to make it just right here. The investment has been stunted, as you know. There have been very few, no really BLA approvals for anything other than cord blood products. And the BLA right now is about a 15-year development pathway for clinical research and about $1 billion investment. There are over 5,000 ongoing U.S. trials, with very few making it past the IND stage. So in the interest of time, I'm going to just summarize what this legislation does. Again, copies of these overhead should be in your materials, or if you give me your card at the end of the session, I'm happy to send them to you, email them to you. So the specifics of what you're going to see passed by the Senate, hopefully even as we speak, is that a new expedited pathway is being created. It's an existing pathway, but it's being applied to regenerative advanced therapies. What is a regenerative advanced therapy? It includes regenerative medicines that are advanced because they're either intended to treat a serious or life-threatening disease or condition, and they have the potential to meet unmet medical needs. This is traditional criteria that FDA has applied and has been flexible in the past. For instance, a serious disease can include a disease that's chronic and long-term, and if it's not treated, it can get worse, which theoretically could apply to orthopedic injuries and other things. The unmet medical need has been defined broadly as therapy that maybe has less side effects. You know, for an example, artificial hip is a surgical procedure that includes anesthesia, but if you can use adipose tissue in the knee, there are less side effects, and that can be defined as meeting an unmet medical need. So this is discretionary on the part of FDA. The only way this regime is going to work is through continued cooperation with CBER, and Dr. Witten and Peter Marks have been very innovative and flexible in how some of this language can be interpreted, and have cooperated through technical assistance to try to make it a workable regime. As I said, guidance on expedited programs, and there's a guidance document referenced here that's online on the FDA website, is flexible on how it defines serious disease or conditions and unmet medical need to apply a little bit more broadly than you might imagine. Regenerative medicine therapy includes cell therapy, therapeutic tissue engineering products, human cells and tissue products, and combination products using those therapies. It does not include gene therapies. That may be the next generation of legislation, but there are pluripotent technologies and others that may not be defined as gene therapies under certain situations, but that is the scope of what's being enacted. And it doesn't include products regulated solely under 1271. And, you know, the use of 1271 is dependent on the guidances that Dr. Witten summarized, and guidances which we hope will be adapted to stem cells in some form before they become final. Under this legislation, the sponsor must request FDA to designate a product as a regenerative advanced therapy, and that designation request can be made any time after the submission of the investigational new drug application at the beginning of the process. So companies will know before they get to phase 1 whether these expedited pathways are available to them, and that should help with encouraging additional investment and creating a more defined regulatory pathway. FDA is required to decide whether to grant this designation within 60 calendar days of receipt. And if request is denied, FDA is accountable and must include a written description of the rationale for the denial, so other companies coming behind this company can understand how FDA is going to apply the criteria and the tools with which they're being provided. If the designation is granted, the sponsor here is eligible for all the benefits of expedited priority, including priority review, breakthrough drugs, as you've heard in Dr. Witten's speech, which includes interactive meetings and advice by senior managers and cross-disciplinary project leads, efficient trial designs, and other helpful innovations. So accelerated approval is available still at the discretion of FDA, and there has to exist a surrogate or intermediate endpoint that's reasonably likely to predict long-term clinical benefit, or reliance on data obtained from a meaningful number of sites, including through expansion to additional sites as appropriate. Accelerated approval generally allows fewer, smaller, or shorter clinical trials than is typical for a drug receiving traditional approval. So again, this is the first step. It's an experiment to see, using these regulatory tools, whether some of the logjam of trials underway can be broken and can lead to submission of an affordable BLA. Post-approval requirements are included in accelerated approval, as you know, and it's discretionary to FDA how it will require those phase 4 or post-approval obligations, but the FDA toolbox is expanded. It can include real-world evidence, which is the mantra of 21st century cures, and such as electronic health records, patient registries, and other sources, in addition to adequate and well-controlled clinical studies, or larger confirmatory data sets, or post-approval monitoring of patients treated prior to approval. Under this legislation, FDA is required to report to Congress annually the number and type of applications filed, approved, withdrawn, or denied, and the number of applications granted accelerated approval or priority review. So there will be complete transparency that will guide future applications. The specific process is also included for FDA to convene a standard-setting body to try to develop some of these consensus definitions and create regulatory predictability. And FDA will be a full and cooperative partner in the standard-setting deliberations. So, in summary, and we're happy to entertain questions, and sorry about the time here that is short, but again, there are sessions that will amplify and explain some of this, some of these provisions throughout the week here. There's clearly a cell therapy revolution, and you're all revolutionaries. Congratulations. There's a proliferation of attempts, however, to bypass the existing system, and that regulatory balance needs to be preserved. We're trying to create a tailored regulatory pathway with the assistance of many philanthropists. You've heard about Bernie Marcus and Bosarge Life Sciences and the Bipartisan Policy Center, and even ARM is on board with this now as an experiment to try to tailor a regulatory pathway to the unique attributes of cells and tissues. The Congress in Washington recognizes the need, and this is being put on equal level with the cancer moonshot, with biosimilars, with immunotherapy, and other personalized medicine initiatives. So it is recognized as a revolution with a need to expand and modernize. The current language is very modest. It's a first step, but we believe it's justified and it's totally within FDA's regulatory control. So as we said, there's nothing new here. The cooperation needs to continue to exist. It's going to be done case by case, sponsor to regulatory reviewer, but there's a new toolbox, and it's all our hope that FDA will seize these tools and this opportunity to continue to promote innovation and move beyond the status quo. Thank you very much.
Thank you so much. I know we're running a little bit behind time, but if there's one or two short burning questions, we can take those before we leave. I believe the speakers should be around a little bit today if you want to tap on them. We have one question.
I just wanted to see if Dr. Whitten could comment on any possibility of a timeline for when the final guidances on human cellular tissues might be issued.
You're going to have to come up here. I'm sorry.
Yeah, it's
Sorry.
barely worth my coming up to the mic to give this answer.
I don't know.
Yes, well, we're certainly actively working on it, but I really can't provide you any kind of time frame except to say that we'll do it as quickly as possible. I mean, unfortunately, that's just, the nature of finishing guidance documents, but I can say we're actively working on it now.
Okay, thank you.
One more question.
Any thoughts on the exclusion of 361 products in the Cures Act? Anyone?
Mark, is there any exclusions? Can you say that again?
And any insight into the exclusion of 361 products in the Cures Act?
There aren't bright lines here, and the hope is that 361 will be adapted or expanded, and some of these guidances that include specific provisions excluding adipose tissue use, will focus on the cell itself and its biological characteristics and not the underlying tissue from which it's harvested, which FDA has the ability to do under the existing regulations. So, it's a work in progress. Yeah.
Thanks.
Thank you. I need to take one last question. Arnie Kaplan, or he won't talk to me after the meeting. Go ahead.
Mark, so what do you see as the next wave of change or legislation that is going to need to be passed to improve upon this particular legislation, gene therapy aside?
Yeah, so thank you, Arnie, and for your contributions to this magnificent day here. I think the 21st Century Cures Act was a collective effort of many, including FDA, in trying to formulate something that acknowledges existing pathways, but can move forward. Part of this is dependent on, whether the glass is half full or half empty, and how some of these first phase, first INDs and first phase 1 and phase 2, trying to move through this pathway, move. maybe there's a need to do nothing further, and the ball is clearly in FDA's court with their experience and expertise. There are, however, subsequent waves. In Washington, we have a new administration. We're likely to have a new FDA commissioner who also is in his senior years and is a billionaire and has some benefit from cell therapy in the future for his own physical condition. But PDUFA comes up next year, so the funding of a third of FDA is funded by user fees. Generic drugs, innovator drugs, branded drugs, animal drugs, medical devices, it's all paid for by user fees, and that's a must-pass legislative package that'll probably move through in September, October of 2017. So that may not be enough time to see whether this legislation is successful and how it's implemented by FDA, but there are always second chances.
Thank you very much. Thanks for attending the session, and enjoy the rest of the conference. Thank you.